Hello,
Hello.
hello, hello. Is my sound clear to all of you? Am I audible to all of you? Is my voice clear? Okay, so we shall start with the electrophoresis now. Today's topic for the electrophoresis is actually gel electrophoresis. Okay, so let's begin with it within a fraction of second. Okay. Okay, so welcome all of you in the today's video. So in today's video, we are going to see what is gel electrophoresis and how different components of gel electrophoresis are there that we are going to see in today's video. Okay, so first of all, is the video visible to all of you? Okay, so we shall start with it. Okay, so let me start with the today's lecture. In the today's lecture, we are going to see the gel electrophoresis. Okay, so first of all, let's start with the topic. Let's see what is electrophoresis. Electrophoresis is, remember, always the movement of charged particles. Okay, in a particular medium. So here the influence of electric field is also very important. Plus there is an involvement of different moieties that are being moved in the electrophoresis. Okay, so let's start one by one. So electrophoresis is a method where charged molecules in solution, chiefly proteins and nucleic acid, migrates in response to the electric field. Okay, so now here the charge molecule present on the particular charge present on the molecule is determined by the different groups that are present in the molecule. Okay, the charge may be positive or negative. Okay, so if the positive charge is there, then the particles is going to migrate towards the negative charge, and if the charge molecule is having negative charge, then the particles are going to migrate towards the positive charge. Okay, so let's see what are the different moieties that are present that moves. Remember, whenever we separate DNA into the electrophoresis, DNA is having the negatively charged due to the presence of phosphate group, so it moves towards the positive charge in electrophoresis. Okay, so we can do the electrophoresis for a number of molecules such as DNA and RNA. Okay, so let's see the rate of charge or the rate of migration or the speed of migration in the this electrophoresis is dependent on the electric field that you are applying. Okay, more the electric field, more will be the faster will be the movement of the charge. Okay, plus it also depends on the size and shape of the molecule, and it also depends on the size and shape. If the molecules are larger in size, then it will lag behind, or it will move slowly, and if the molecular size is more, it will move faster. Okay, then it also depends on the viscosity of the buffer that we are using. Now electrophoresis, there is an electrophoretic buffer in which this the movement of charge is happening. So ionic strength, the viscosity and the temperature of the medium in which the electrophoresis is done is also very important here. Remember, the electrophoresis is always done in a specific matrix. Okay, there is a gel in which the electrophoresis is done, and based on the type of electrophoresis which you are performing, different kinds of gels are there. Okay, so let's see in details about this topic. Okay, so electrophoresis is a very important analytical tool in which you can determine lots of things about the biological molecule. Let's see what are the different types of gels that are used in electrophoresis. Okay, so now the gel that are prepared in electrophoresis, this can be in the form of tubes, slabs, and flat beds. Okay, different types of matrices are there, and it is available in different shapes. Okay, so now we are going to see what are the constituent. First of all, remember there is a matrix, that is a gel that can be made of different kinds of things depending on the molecules that you are separating. Then there is a buffer chamber in which the buffer is poured and gel is casted. There are two electrodes here. Remember, there are two electrodes, one is a cathode and another one is anode. Now the cathode which is present is always negative in charge, and anode is always positive in charge. Okay, so cation, since it is positively charged ions, the respective cathode that is negative in charge. Okay, so always anode here will be positive charge and cathode will be negative charge. So this is all present in the chamber, that is the electrophoretic chamber in which electrophoresis is done. Okay, so let's see first about the DNA gel electrophoresis. Okay, so agarose gel is used for DNA electrophoresis. Okay, so agarose, what is it? It's a disaccharide made up of two components, that is galactose and 3,6 anhydrogalactose. Now this is the structure shown here is of galactose, and this structure is called 3,6 anhydrogalactose. Okay, so now this molecule is first of all taken in water and heated. Okay, now when it is heated, it starts forming a polymerization like this. Okay, it starts forming a polymer like this. Okay, so now more the concentration of agarose, less will be the pore size. Okay, so remember, the pore size of the agarose gel is inversely proportional to the concentration of agarose. When you increase the concentration of agarose, the pore size will decrease. Okay, so you can see the two different types of gel network that is formed by agarose. This is with the low concentration of agarose, and this is with the more concentration of agarose. Okay, so the pore size in the less concentration of agarose will be less or more, sorry, the pore size will be more here, and in this case the pore size will be less. Okay, now when this matrix is heated and it is allowed to cool, then it forms the three-dimensional gel network in which the DNA molecules can be separated. Okay, based on the molecular weight and its charge. Okay, so now generally the melting point of agarose is 52 degrees Celsius, but it depends on different kinds of agarose, different quality of agarose. It has different varying melting point. Okay, so depending on the application, what kind of separation you are doing in the agarose gel, the gel may be of different types. Okay, now let's see, this is the DNA structure. You can see here there is a nucleotide molecule, then ribose phosphate moiety, and then there is ribose phosphate moiety here. Okay, the negative charge of the DNA is due to the phosphate group, you know it. Okay, now the movement will always take place towards the positive charge. Why the movement will take place towards the positive charge? Because DNA is negatively charged due to the presence of phosphorus. Okay, so this is the rough structure of DNA. So now it will always move towards the positive side, and this will be the negative electrode in the electrophoresis. Okay, this is how the three-dimensional network of agarose is going to form, and DNA will form a band. It will move towards the positive end. Okay, now DNA is not having any kind of fluorescence of its own. So in this case, you have to put a fluorescent dye into the agarose. Okay, while boiling the agarose, after boiling, when the agarose is slightly cooled, you have to add a dye called as ethidium bromide. You can use other dyes also, but generally ethidium bromide is used. Other dyes are also there, such as acridine, orange, and all. These dyes can be used. This dye helps to visualize the DNA under UV. Okay, when you see this gel under UV or UV transilluminator or gel documentation unit, there the UV source is used, And now this ethidium bromide dye, which is added into the agarose gel, it interchelates into the DNA strands when they are moving. Okay. Now this DNA is easily visualized when you put this DNA or put the gel into the agarose gel. Okay. Sorry, UV transilluminator. Okay, so this DNA is easily visualized. Okay, so you need to put ethidium bromide into the agarose gel. Remember, this ethidium bromide is a carcinogenic chemical, so it has to be added very carefully into the agarose gel. Okay, this is how the electrophoretic chamber looks like. Here, this is the gel that is being casted at the middle. There are two electrodes here, and the upper unit is the power pack. Okay, so the power pack provides the electricity, and the cathode is connected and anode is connected. So now here, different units are there. Always remember that these are the wells into which the DNA will be loaded. These wells are casted when the agarose gel is poured. Okay, there is a special casting assembly in which first of all a comb is kept, which forms the well, and then gel is poured. Now when the gel solidifies, the comb is removed and you can get the wells of defined shape as you want it. Okay, now this gel is kept in this electrophoretic chamber. Remember, the electrophoretic chamber is available in different capacities. I am going to show you the photos of different kinds of electric chamber. Flat bottom, vertical, tube electrophoretic chamber are there. I am going to show you the photos here. And the electricity is connected and the electrophoretic buffer is poured onto this. Normally in agarose gel electrophoresis, the buffer which is used is tris acetate EDTA, TAE buffer. It is tris acetate EDTA. Okay. So now this buffer is poured into the electrophoretic tank that is responsible for electrophoretic mobility. Okay. And the cathode is anode is connected. Generally in agarose gel electrophoresis, at the starting, the voltage is kept at 50 volts till the bands of the DNA moves into the gel. Okay, once the band of the DNA moves into the gel, okay, and once it happens, the voltage is adjusted to 100 volts, and it takes around 1 to 1.5 hours for electrophoresis to be complete. Generally, we move the DNA bands to the 2/3 of the gel. It depends on the type of DNA which you are moving. So accordingly, you can put your time interval for the electrophoresis. Okay. So now this is the positive charge and this is the negative charge, and DNA is going to move towards the positive charge, that is the anode. Okay. Okay, now this is a vertical chamber I am showing you here. One of the example of electrophoresis. Generally, the vertical chamber is used for protein electrophoresis. Even for agarose, also vertical chambers are there. You can see there are two chambers, top chamber and a base chamber. Okay. So now in the top chamber, the electrophoretic buffer is poured and there are well present in the gel here. Okay. The below coil, the coil here in the electrophoretic chamber is generally made up of nichrome wire. Why? Because it is inert, it is resistant to the buffer and the gel. The chemicals that are present in the buffer and the gel, they are resistant. The coil is resistant, so the wire used here as an anode and cathode is platinum wire. Remember, platinum is used here. Okay. So now here you can see the two terminals are then connected to the power supply that I showed you, and now the gel is kept here and the sample is being loaded. Now the sample is loaded via micropipettes, and once the sample is loaded, the gel cover is kept on that, and then electrophoresis is proceeded. Okay, and you put the current supply. You can see the bubbles in the wires, that is platinum wire that is placed over here and here, that is the positive as well as negative electrode. Okay, so now you can see here the cathode is present and anode is below. Always remember, anode is positive in charge, and when you use, okay, so when you use different molecules to be separated, that molecules must have a negative charge. Okay, so if protein molecules is there, protein molecules can have different charge. Okay, there are certain positively charged proteins, there are certain negatively charged proteins. For negatively charged proteins, it can easily move into the agarose gel. Okay, because it will move towards the positive end. But for positively charged proteins, they have to be given the negative charge. So in such case, the proteins are supplied with the negative charge using the molecules such as SDS, that is sodium dodecyl sulfate. Okay, So it is an anionic detergent and it gives the negative charge to the protein molecule, so that they can be easily separated into the electrophoresis assembly. Okay. So remember, the movement which you want, that opposite charge should be there on the biological molecule, such as the DNA and protein should have negative charge when it has to be separated or it has to be moved towards the positive charge. Okay. So you can see here, in case of proteins, there are different kinds of gels. Okay, I will go into the details of this also. And there are different kinds of gels such as stacking gel and separating gel. Okay, I am going to elaborate upon that also. In protein separation, there are two different types of gels that are used. First is stacking and other one is separating. Okay, so we are going to see that in details after this. Okay. First of all, let me show you the different types of gel electrophoresis unit, not agarose, but other electrophoresis unit. This is a tube gel electrophoresis unit. You can see there is a electrode on the top, and these are the tubes that are present, and this is the chamber, and the inner chamber is kept in a outer covering like this. Okay, the covering is also having the buffer in it. Okay, and the electrode is connected below, and the upper electrode is connected onto the top like this. Okay, and the electrophoresis takes place in these kind of tubes. Sometimes these tubes can be very small as capillary in nature, and sometimes they are bigger also. It depends on the type of electrophoresis that you are doing. Okay, it comes in various chambers. You can see the vertical chamber is of different sizes. Okay, it depends on your use, what kind of use you are doing. It is present on different sizes. Okay. This shows a vertical chamber, electrophoretic chamber, that is generally used for SDS page. Okay, you can see here vertical chamber. It is showing you. Okay, this is the gel casting. This is the comb in which the gel is casted. You can see here comb, and this is attached to a gel slab assembly or gel casting assembly. There are two plates in which the gel is prepared for SDS page. Okay, yeah, flat bed unit is another type in which the gel is created in a flat bed. Okay, there is a specialized casting tray for this, and the gel is poured onto this casting tray. Okay. Okay, so yeah, let's see the current kinetics here. You can see the electrophoresis surely depends on the Ohm's law. Okay, which is V is equals to IR. Here E is electrical field. Now as the voltage increases, the current flow increases. So V is directly proportional to I, that is voltage is directly proportional to current according to the Ohm's law. You know it. Okay. So as you increase the voltage, the current also increases and the heat also increases. Okay. So current is inversely proportional, directly proportional to heat. Remember, as the current flow in the electrophoresis unit increases, heat also increases. Okay. So you have to always take care that you will not exceed certain voltage. For example, for biological molecules such as protein and DNA, the voltage should not exceed 100 volt, because the current increases and thereby increasing the heat. So as the heat increases, it can melt away the gel. Okay, so your electrophoresis will not be proper if the gel gets melted. Okay, so therefore you have to maintain an optimum temperature by maintaining an optimum voltage, which will in turn maintain an optimum current in the agarose gel. Okay, so this is the formulas that are here important, and the parameters such as electric current, voltage or power is always held constant in this case. Okay. I already told you that if you are not heating, you are not providing a constant electricity, then heating is done. So to keep the heating constant, a specialized power pack is used, that is the machine supplying power, which keeps the current constant. Okay. So the net charge present on the protein molecule is also dependent on the pH of the medium. Okay. So this is special. This is especially important in case of protein molecules if you are separating by electrophoresis. Now, why proteins are amphoteric in nature? Okay, it can be a positive charge or negative charge also. Okay, so therefore, you should realize the type of charge that is present onto your protein which you are separating. Okay, so depending on the number and the kinds of amino acid it is carrying, the charge is different. If the number of basic amino acids is more, the protein is having a positive charge, and if acidic amino acids is more, the protein is having a negative charge. and if the protein is neutral, then it will not move in an electric field. So all in all, they have to be given a negative charge in for which it has to be electrophoresed. Okay, I already told you the negative charge is given by certain detergents such as SDS. Okay, so important parameters at every stages of electrophoresis. First is the polymerization. Now, when you are preparing the gel, it has to be prepared very carefully. Okay. So why? Because it's an exothermic reaction. Since the gel is prepared or the gel is polymerized, it gives out heat. Okay, there may be certain irregularities in gel that can be there. If there is a bubble in the gel, then it can create it will not form a uniform gel. Okay, so there is an irregularity in the gel, so that has to be avoided by using a proper concentration, avoiding the bubbling in the medium. Okay, then the pore size is also important for gel. If you increase the concentration of the the matrix, that is agarose, or it can be page, that is made from acrylamide and bisacrylamide. Page is polyacrylamide gel electrophoresis. This gel is used for separation of DNA as well as RNA. Now, this the concentration of polyacrylamide or bisacrylamide, acrylamide, or agarose determines the pore size. Okay, so you should be very careful while selecting the concentration of agarose or acrylamide or bisacrylamide, because the pore size is going to depend on that. Okay, now in electrophoresis, you have to take care that your protein should not denature. If heating is over, or heating is done over, or more amount of heating is done, as the voltage or current is more supplied more, then your protein gets denatured and it is not going to separate properly. Okay, so therefore you have to maintain a constant voltage. Okay, and then you have to avoid smile effect. Okay, for example, as I showed you in previous slides, I'll show you again what is a smile effect. So you can see here, suppose if the proteins are moving and the electrophoretic field is in more in between. Okay, so now the DNA bands are present at the periphery or sides will lag behind, and it is going to create a smile like this. Okay, so the bands will form a smile like this is called as a smile effect. Okay, so the wire, platinum wire, which is acting as an electrode, should be uniform throughout, and it should provide a uniform electric field so that all the bands are moving in a proper direction and a proper speed. Okay, so so therefore the smile effect should be avoided by taking a proper electrodes or the uniform size of the wire that is present. Okay, then temperature regulation of buffer should be done. Normally the TAE buffer or the electrophoretic buffer, that is tris glycine buffer, tris glycine SDS buffer, or other electrophoretic buffer that are used, are helping in maintenance of temperature as well as the pH of that solution. Okay, so therefore the buffer presence is also very important. Okay, what is the role of solid matrix? Solid matrix such as agarose, acrylamide, or bisacrylamide, this helps in separation. It inhibits convention and diffusion, which would otherwise impede the separation of molecules. So all the kinds of diffusion and conventions, currents are nullified by this matrix. Okay, it allows a permanent record of the result through the staining after run. Okay, when you run the gel, you can stain that gel also to keep a permanent record, or you can take a photograph of that particular gel after analysis if you are having a gel documentation system, or if you are having a UV translaminator for DNA, you can just take a photograph. And it provides the additional separation through the molecular sieving, because since the pore size can be greater or smaller, you can separate the molecule based on the charge or based on the molecular size also. So the matrix here is very important. Okay, there are two types of matrix that are generally used. I already told you, that is agarose and polyacrylamide. Okay, this forms a porous gel network that helps in separating the different biomolecules. Okay, generally agarose is used for DNA separation, and polyacrylamide gel is used for protein separation. But in some cases, the polyacrylamide gel made from acrylamide and bisacrylamide, that are the monomers in this gel, can be also used for DNA separation. Okay, so what is agarose? First, let's see. Agarose, it is highly purified polysaccharide, from which agar agar is a polysaccharide from which the agarose is isolated. It is isolated from a brown algae, that is rhodophyce. The name of the algae is Gelidium corneum. Okay, Gelidium corneum. This is an algae from which the agarose is isolated. The melting temperature can be as low as 40 degrees Celsius. Generally, the agarose gel that is used for educational institutes and all, it is having a melting temperature of 52 degrees Celsius. But there is low melting agarose also required for very specific applications in which DNA has to be recovered from the gel. Okay. So the pore size, I already told you, depends on the concentration of agarose. Okay. So these gels are fragile. However, they are actually hydrocolloids and they are held together by the formation of weak hydrogen bond and hydrophobic interactions. Okay. So you can see the basic disaccharide repeating unit of agarose, that is galactose and 3,6 anhydrogalactose. You can see the connection between them. This is the anhydrogalactose molecule and this is the galactose molecule here as shown in the figure. And at solid state it is like this. When you are heating it, it starts polymerizing and the final gel structure becomes like this. Means this kind of pores that can be created into the gel. The pore size will be very small if the concentration of agarose is taken in a large amount. Generally, the 1% concentration of agarose is taken. So for 50 ml, you have to take 0.5 gram agarose. For 100 ml, you have to take 1 gram agarose. But in some cases, for example, if you want to separate a plasmid molecule, which is very small in size, you have to take a low pore size agarose. So the concentration of agarose taken here is more for the plasmid isolation, especially. Okay, 0.8% or 1.5%, it can be depending on different, separations. Now the polyacrylamide gel, these gel are tougher than agarose gel as it is formed by two different monosaccharide, that is acrylamide and bisacrylamide. Okay, it forms the cross linking. Acrylamide forms the straight, parallel linkages. Bisacrylamide forms the linkages in rows. Okay, you can see the reaction of acrylamide and bisacrylamide, and it forms a gel like this. Okay, now for the formation of gel in case of acrylamide molecule, there is a very important requirement, that is persulfate molecule. Requirement of persulfate molecule is there. Okay, so while preparing a polyacrylamide gel, sorry, you are using a chemical such as ammonium persulfate. Now this ammonium persulfate helps in polymerization. Okay, now this ammonium persulfate polymerization is a very slow process. Okay, so while forming the agarose gel, obviously you have to take acrylamide and bisacrylamide. Generally the bisacrylamide taken is methylene bisacrylamide. Okay, the structure is shown to you here. Ammonium persulfate is added into the reaction mixture and initiates the polymerization, because the ammonium persulfate provides a sulfate radical that is actually required for polymerization. Okay, now when the polymerization is starting, the polymerization is actually very slow. Okay, so if you wait for this, it will take a longer time. So for that purpose, you add timid. Okay, timid is a chemical which is added that acts as a catalyst. Okay, catalyst is what? Which fastens the reaction. So here the polymerization of acrylamide is made fast due to the addition of timid. Okay, so generally ammonium persulfate and timid are added in equal amount or 1:2 ratio. It depends on what kind of gel or what strength of the gel you are forming. Okay, so always in protein, there are two types of page. It can be a native page or a SDS page. Okay, so in native page, what is happening? There is only one gel. Okay, but in SDS page, there are two gels. Okay, so while preparing the page, that is polyacrylamide gel electrophoresis, the total percentage of acrylamide and bisacrylamide is taken, or is very important, which forms the cross linking. Okay, and the presence of oxygen should be removed from the mixture because it will hamper the gel formation process. There are two ways in which the polyacrylamide gel can be formed. First is chemical method, the method that I told you just now, by the addition of persulfate radical and timid is a chemical method. It can be also done by photochemical method. Okay, so you just add acrylamide and bisacrylamide in certain percentage which you require, and you add a chemical such as retinol, that is vitamin A. Okay, you put that chemical and put the gel under the sunlight. Okay, so due to the presence of sunlight, there is a photochemical reaction which produces the retinol radical. Okay, it will form a retinol radical, and this radical will do the polymerization. Okay, so you need not to add ammonium persulfate or timid. In that case, you can do the polymerization by photochemical method also. Okay, so this is an alternate method. Okay, so after that you can go for autoradiography. After the electrophoresis, you can do the analysis of gel by autoradiography, and then you can go for densitometry. Or either if you are using a capillarity, you can use for detector. You go for detector, or if you are going for hybridization, you can do the hybridization by autoradiography or immunodetection. Okay. In SDS, I told you about native page. In SDS is used to give the negative charge to the detergent. Plus, it also breaks the disulfide bridges. Now, for electrophoresis of protein, especially, the protein molecule needs to be in a primary state. Okay, so all kinds of interactions in the protein which is forming secondary, tertiary, or a quaternary structure has to be removed. So SDS along with beta mercaptoethanol, that is used in a gel loading dye, that is sample loading dye for protein, removes the all the secondary structures and all kind of 3D structure of protein, and makes the protein in a primary structure, that is a linear polypeptide form. Okay, so that it can move easily into the gel. Okay, so now therefore the sodium SDS is used to give the charge to the molecule. Okay. Okay, now you can see how treatment with SDS and mercaptoethanol changes the 3D structure, that can be secondary, tertiary structure, into a primary linear form, and this protein becomes negatively charged due to the coating of SDS molecule, and it separates easily onto the agarose gel. Okay. Continuous and discontinuous buffer system. The buffer system can be continuous, means what? The diagram which I have shown you at the starting was an example of discontinuous buffer system. It can be a continuous buffer system. Into the continuous buffer system is the one in which the buffer is kept poured onto the whole gel. Okay, discontinuous buffer system is one in which the buffer is kept at two ends, and in between there is a gel that is covered by glass plates. Okay, so you can see here, these are the two types of buffer system, and these are the two types of gel. Here you can see this is a gel which is made from a single concentration, that is a resolving gel, or it is also called as separating gel. Okay, here the buffer system is kept separate at the starting, that is the cathode end and anode end. This is discontinuous buffer system, and if the buffer is kept as a whole for whole gel, then this is called as continuous buffer system. Okay, now the gel is also a continuous or a discontinuous one. You can see here the gel is actually a whole gel is made from same concentration, so this is called as a resolving gel. Okay, and it is also called as native page, where the resolving gel is used. Okay, other one in which the proteins are separated generally uses two different types of gel. The first one is stacking gel, and the second one is resolving gel. Okay, remember the stacking gel is having a different pH, and resolving gel is having a different pH. Generally the stacking gel is having a low concentration of acrylamide as well as bisacrylamide. The resolving gel is having more concentration of acrylamide and bisacrylamide. Whatever the protein molecule that you are loading here is going to stack at this level. Okay, the protein molecules are going to stack. Okay, and when it enters the resolving gel, due to the pH difference and the molecular difference, molecular size difference, the proteins are going to resolve or separate onto the resolving gel. Okay, so this is how the separation of protein molecule based on charge, based on the molecular weight, takes place in SDS page gel electrophoresis. Okay, I'll show you just an example of SDS page. You can see here beta galactosidase, then cat protein, MPB protein, and green fluorescent protein separation is done. Okay, the separation is done on the basis of molecular size. The protein bands are normally not visible. See, in case of DNA, for DNA to be visible, we add ethidium bromide like chemicals. Okay, but in case of protein molecules, you have to use different stains after the electrophoresis is done. Now this staining is Comasi brilliant blue staining. Okay, this is a blue colored dye which is used for staining. Okay, That is a chromacy brilliant blue. Otherwise, there are also alternate strains available. Other strain that can be stained, that can be used, is silver stain. So when you use a silver stain, the bands look like this. Okay, the bands looks brown in color, and the separation takes place like this. Okay, you can see the SDS page gel that is being separated onto the SDS page gel. Okay, so you can plot the graph also depending on the mobility and the molecular weight, and you can determine the molecular weight of different protein molecules. The graph paper that is used here is generally semi-log graph paper. Why? Because one of the axis of the semi-log graph paper is a normal axis, and other axis is logarithmic axis. Okay, therefore it is a semi-log graph paper. Okay. Now here the most important thing is the RF value. Okay, so when RF value, what is it? Is a linear relationship between the movement of bands. Okay, remember RF value is calculated by the formula length or the distance traveled by dye divided by distance traveled by band, protein band, divided by the distance traveled by dye front. Okay, so you can see in this gel the dye is going to move at this level. Okay, you can see here there is no presence of dye because dye has moved further. Okay, so dye will move to whatever the sample loading dye that is present in the sample will move to a certain extent. Okay, so you have to, from the well, you have to measure the length of the or the movement distance of the dye traveled. Now you have to measure the distance of the band. Each band's distance you have to measure. Okay, now when you divide the distance traveled by band, DNA band or a protein band, divided by the distance traveled by the dye or dye front, you get the relative flow or RF value. Okay, now this RF value is plotted onto this graph. Here the RF value, and here there is a molecular weight. Okay, you can see the molecular weight increases in 10 folds. Therefore this axis is logarithmic axis and this axis is a linear axis. Therefore this graph paper is called as a semi log graph paper. Okay, now after the electrophoresis, if you have to go for detection, okay, so you can go for blotting techniques. Okay, so first blotting techniques was invented by scientist E.M. Southern in 1975, in which suppose you have done the electrophoresis of a particular sample onto the agarose gel, and you want to detect a particular band or a particular gene if for the presence of that gene in that particular DNA. Okay, so you can go for hybridization. Southern hybridization, northern hybridization, and western hybridization are certain techniques. Are the three techniques where DNA, RNA, and proteins are hybridized or detected respectively. Okay, these three techniques are discovered by different scientists. The first technique of hybridization was discovered by scientist E.M. Southern in 1975, in which the DNA hybridization is done onto the agarose gel. Okay. So you can see this is the electroblotting assembly. Now what happens in electroblotting? Now see, after electrophoresis, you cannot store the gel for forever or for lifelong. You cannot store the gel. Okay, so it has to be blotted onto a particular paper membrane, that is generally nitrocellulose membrane or nylon membrane. Okay, so you cannot keep the gel as a record forever. Okay, you can keep the membrane as a record forever. Okay, membrane, it is a paper that is a nitrocellulose paper or nylon membrane paper. In case of northern blotting, the paper is diazotized paper, that is amino benzoxymethyl paper. Okay, so here you can see how the sandwich is done into the electroblotter. Now the after electrophoresis, the bands are moved into the agarose gel. Now these bands has to be casted onto a membrane. Okay, so this kind of sandwich is done. You can see here gel holder on the both sides. Then there is a sponge pads present in the both sides. Then there is a filter paper. After that, there is gel, and then there is a transfer paper. Transfer paper can be membrane, that is nitrocellulose or nylon membrane for DNA. It can be used for protein separation also or protein blotting also. And now when the membrane when the membrane is blotted with the bands, then this membrane can be now fixed. The whatever the bands that are formed on the membrane or blotted onto the membrane by the electroblotting assembly. This is the electroblotting assembly. It looks like this. Generally, it is a vertical assembly where two electrodes are present. See here, one electrode, here one electrode, and this is the gel. So generally this side will be the positive side, positive electrode, and the bands present in the gel will be blotted onto the membrane, okay, by the electrophoretic mobility, okay. Now this membrane, which has the DNA bands or protein bands blotted onto it, is now immobilized by baking or UV cross-linking or other methods, okay, and it is washed by buffers, different buffers, and after washing, the DNA can be detected by using southern hybridization, northern hybridization, or western hybridization by either radio label detection or non-isotopic detection, okay. Generally, the separation also takes place in depending on the isoelectric pH in SDS phase medium. Okay, you can see how the proteins are separated based on the charge in the SDS phase gel. Okay, so isoelectric focusing is also very important, phenomena in which the protein bands are separated depending on the pH. At specific pH, specific protein bands are separated, and it depends on the amino acid constituent of that particular protein. Okay, the gel separated can be a 2D gel, 1D gel, 2D gel electrophoresis. Here, the electric field that is applied is in a XY coordinate manner, and you can see how the gels are separated and how a 2D gel looks like. Okay. This is a 2D gel electrophoresis where proteins have migrated to different areas. This is the basic side, and that is the acidic side, and this is the SDS strength which goes on decreasing. Now the separation of protein takes place like this. You can see the images from yeast, images from rat kidney. Yes, differential gel electrophoresis you can also do. In case of differential gel electrophoresis, you are using two-dimensional electrophoresis, but the thing is you are using two different dyes. Okay, you are using two different fluorescent dyes. It can be 2 or 3 dyes that can be used for separation. Okay, see here 3 dyes. I have just listed an example here. In case of Amsham system, so cis 2, cis 3, and cis 5 dyes are used. You can see the different dyes are attached to the proteins, and like this the different band separation or detection of different bands can be done. Okay, so this was all about the gel electrophoresis that I had to take in today's lecture. Thank you for joining the today's lecture. If there is any doubt, please do comment in the comment section below. Thank you for joining, all of you.